public health implications of traumatic brain injury and Alzheimer's disease are immense.
Over 7 million older Americans suffer from the brain and memory destroying illness of Alzheimer's disease. That number is going to double with the increase in the older population in the coming years. Traumatic brain injury is most often an affliction of younger individuals who have suffered a sports injury, individuals who were in the armed services and suffered an injury. They're smaller in numbers, but they're losing an entire life ahead of them.
This is, of course, a strain in terms of human suffering for the patients. It's also a strain on their caregivers, and of course, it's an enormous financial strain on both the patients and their families and society as a whole. I do research on neurodegenerative disease.
I'm a physician scientist. I do basic medical research, and now more broadly in drug development.
The blood-brain barrier it's essential for lifelong brain health. It's a network of cells that sit at the interface between the cardiovascular system and the brain, so that the brain is protected from various toxins in the blood that it shouldn't be exposed to. The second thing the blood-brain barrier does is it actively eliminates toxins from the brain by pushing them out into the blood. And then the third thing that the blood-brain barrier does is very precisely couples the delivery of blood to the various parts of the brain when they need it. As various degenerative processes occur with the blood-brain barrier, such as with an injury or in a disease like Alzheimer's disease, all three of those functions of the blood-brain barrier become impaired. The main finding of our paper is that we discovered a way to protect the brain under conditions of injury as well as genetically driven Alzheimer's disease by protecting the blood-brain barrier. Specifically, what we found is that there's one enzyme, it's called 15-PGDH, that is highly enriched in certain cell types that migrate towards that blood-brain barrier.
What we found was that increased 15-PGDH primes inflammatory cells to damage the blood-brain barrier. That when damaged, amplifies, magnifies, and perpetuates inflammation and damage to the brain. And that by shutting off this enzyme, we could block the entire inflammatory and damage cascade. And we found we could do that with a drug that was developed in my lab that was so potent that it worked even when diluted down to 1 part in a billion.
One of the original observations when Alzheimer's was discovered was these prolific accumulation of amyloid plaques all over the brain. And so it's always been thought that eliminating those plaques might be a way to treat the disease. And we showed that when we inhibited the 15-PGDH enzyme, that we didn't affect amyloid pathology. So even though they had amyloid plaques all over their brains, they didn't have any of the cognitive impairment and other symptoms that they would normally see in this model of Alzheimer's disease. I think the novelty, the non-canonical aspect of our work is that we showed that protecting the blood-brain barrier is itself sufficient to preserve mental and cognitive function in these models of traumatic brain injury and Alzheimer's disease, and that was not expected. Our major focus is, will this be something that can help patients? And so we are actively working to try to get this into clinical trials for these conditions of brain injury and disease. In other words, can we use this as a launching pad to learn new things about the brain that can help us identify additional complementary ways to protect patients in the future?
Andrew's lab, with their wonderful understanding of brain disease and the blood-brain barrier, and my team with their understanding of how to work with PGDH and how it worked to change the behavior of cells. It's a beautiful demonstration of the value of collaboration.